Well, thank you very much, Pat. Probably one of the best introductions I've ever had, and I really appreciate it. It's really quite an honor and a privilege for me to be here today. I'm very excited about sort of what you're all doing and how we're working with you, and I'm going to talk a little bit about some of the efforts that we've undertaken so far to try and move the ball forward to actually develop a drug for CMT1A. So some of you may be familiar with this, and this is the timeline and also the attrition of what happens during drug discovery. And as you can see, we start out in the very early phases of our work when we start working in a pharmaceutical company at around 1 million different compounds that then we proceed through successive sort of steps where we sort of run them through assays and models, and we optimize the molecules so that they become safe and effective drugs. And then we move those into clinical trials like we heard about earlier from some of our physicians here. And then ultimately, if we're really, really lucky, we end up with a drug that becomes approved. But as you can see here, in terms of the timelines, for some of the early work, it can take many years, 6.5 years, and then 6 years for clinical trials, and then another year to sort of work through the process at the FDA to actually get approval for a drug so that it can be made to our patients and they can take it to start treating their disease. But one of the things that really sort of changes this whole paradigm is the opportunity to sort of partner and leverage sort of existing capabilities from other parties to really help accelerate both in the development of assays, the design of the clinical trials, and also dealing, potentially with the FDA. And so these partnerships really do sort of increase our opportunities to increase the probability for success and decrease timelines. And as Pat mentioned, we recently had a partnership that was signed last year with the CMTA and Sanofi-Genzyme in search of a cure for CMT1A. And together, we really do feel that we are trying to make a difference for CMT patients. And this type of partnership is extremely important. And, I think people like Pat should be, sort of significantly commended for their vision and the approach they've taken to sort of put in place, a partnership that greatly facilitates our ability to develop a drug for you. This is drug discovery process is an enormously challenging process. It's difficult. It's fraught with many failures as one goes forward. And knowing that we have backup in the form of the CMTA is incredibly helpful to us on a number of different fronts. And so what the CMTA has done has been extremely beneficial for us. So I just want to sort of highlight, sort of how long this process has taken and some of the things that have been involved in sort of getting us to the point where we were, and highlight some of the important aspects of a partnership. So, key to a partnership is, having a strategy and vision, and that comes from executive leadership like we have from Pat, but also, sort of scientific capabilities like what you heard Pat talk about with the STAR network and scientific leadership. And this is where Mark Scheidler, who's sort of been leading sort of the effort on the scientific side with us, has been extremely helpful, and this is coming from the CMTA. And what's really important is if you look at this timeline and you see sort of some of these yellow dots along the way and the green dots, these are meetings and events that we've had to sort of get us where we are today, starting with some of the discussions with the CMTA in 2012. The red dot is when we had a reorganization at Sanofi that slowed us down for about 6 months. But the green dots are where big things happened. Meetings in Arizona, commitments from the CMTA, commitments from Sanofi, until we actually get to execution. And so what's the important part here that I want to emphasize is this commitment and perseverance and having that sort of patient perspective that you all bring to the table to us in terms of making us move forward along this trajectory to actually implement a collaboration. Collaboration, doesn't happen, as you can see, in a day, doesn't happen in a week. It took us over 3 years to kind of get to the point where we actually started the work to actually move things forward. So what are the critical components of actually moving a collaboration like this forward? Well, we need to have strong contributions from both parties. And I think what you heard from Pat, before lunch about the STAR network is an incredible contribution coming to any sort of partnership or any pharmaceutical company working on drug discovery, and incredibly helping us move the ball forward. And then from the Sanofi side, there's lots of components there too that we're bringing as well, expertise in drug discovery and drug development. But also the financial support. So people may ask, why do we need to have financial support for a major pharmaceutical company like Sanofi for this to happen? Well, and I've explained this for a number of reasons. Quite frankly, we wouldn't be working on CMTA if it wasn't, CMT1A if it wasn't for the CMTA. That's what it comes down to. And having that vision and the financial support to sort of initiate some of these activities at a very early stage in a high-risk environment enables us to sort of start this activity and move things moving forward to actually discover a drug. But it also does a couple of other things. It commits both parties to a contractual agreement where we're all on the same page trying to discover a drug for this therapy, and we're committed to moving it forward. And then there's confidence on both sides to collaborate. But the most important thing is for my leadership is that we are it's compulsory that we actually fulfill the contractual obligations that we have with the CMTA and move this forward to the next milestone. So these are really critical factors along the lines of supporting the efforts. And other things that are important is a defined work plan with agreed milestones, and that's really key. And that's something that the CMTA worked very diligently. Mark Scheidler with my team and project team leader Paul Wright to really execute and put in place for this. Because we all have this sort of common objective where we all want to get to a point where we have an orally available drug to treat CMT1A. So as I mentioned, there's many strengths and synergies that both the CMTA and the Sanofi-Genzyme bring to the table. With Charcot-Marie-Tooth Association, you have seasoned leadership, as I've spoke about with Pat and Mark. And you have a clear strategy to accelerate research in this area for Charcot-Marie-Tooth disease. And not only that, but you have some of the top scientists in the world who are sort of engaged with this and supporting the activities. At Sanofi-Genzyme, we are a global pharmaceutical company with a global network capable of developing drugs with a proven track record for rare diseases through our Genzyme Rare Disease Unit. We've executed many productive collaborations with patient organizations in the past. But we have a real strong history and capabilities of actually discovering drugs and developing them into therapeutics. And that's particularly so with our team at the Tucson Innovation Center. So our Tucson Innovation Center is located in Oro Valley, Arizona. This is a picture of our site here and location of where the CMTA meeting was, where we actually kicked off this collaboration. This site is the site where the work is being done for the CMT1A discovery work to develop a drug for that disease. So here in this collaboration, we have already probed more than 2 million small molecule samples through the assays that have been provided by the CMTA. The center has significant expertise in employing high-volume chemistry to actually modify and optimize those molecules to actually enhance both the safety and the efficacy of the drugs that we develop. And we also have a lot of biological expertise here with being able to sort of take cellular models, take patient cells and differentiate those to appropriate tissues, motor neurons, for one example, and then hopefully in the future with the help of the CMTA, we're going to be generating Schwann cells from individual patients with CMT1A as well. So we are an integrated lead therapeutic generation unit that really has a track record of innovation and therapeutic discovery. And we are working very hard on discovering a cure for CMT1A. We have robotic capabilities, as you can see here, that we put into play to actually discover that effort. These robots sometimes are working day and night to actually screen molecules in some of these cellular assays. We also have significant expertise in muscle biology, and that's also neuromuscular work as well, where we're sort of creating cellular models that we can employ to recapitulate the disease pathology of interest. As you can see there with rare diseases, Charcot-Marie-Tooth is one of the top projects that we're currently working on, but we work on a number of other rare diseases as well. And so we have a clear idea of what needs to be done and what we need to put in place to move molecules forward. And thanks to the CMTA, we actually have now a number of different cellular assays that have been built in many of the same ways that we would actually build assays to actually move forward into our discovery efforts. This is just an illustration that was actually published by John Sforin and Jim Inglis in 2014 on this assay and the screen that they performed at INCATS, where they've actually precisely integrated a reporter gene at the PMP22 locus in order to monitor for the expression from that promoter, so that we then could identify molecules that would actually bring down the expression back to the wild type. And so we're employing these assays, and we employ those in this system, in this pathway that's shown here. Under the green area is where the collaboration agreement is currently focused. And thanks to, and as I mentioned early on, early in the drug discovery process, it's a lengthy process, but thanks to the CMTA STAR network, many of the steps up front of what we normally need to put in place have been eliminated. And we have the support for additional assays that can be generated to actually help us move things forward even faster. So we're able to execute on the assay that John Sforin had built and Jim Inglis validated at NIH, and move that into our HTS capabilities here, and start moving this towards the milestone here of a lead selection at what we call M1. And so that's our major objective in this collaboration. At the present time, we've successfully completed assay transfer, screen initiation. We've also completed the entire screen, and we're in the process of actually working through sort of some of the hits that we have to try and do back screening and optimize some of the molecules to find those lead candidates that then we'll move forward. And as you can see on this chart here, the next steps that we'll be moving forward into are actually profiling these molecules into additional assays that the CMT STAR network has generated at some of the investigators' labs. So we'll be profiling these in patient cells to actually look that the molecules are actually demonstrating the desired effect. And then ultimately we'll be performing proof of concept studies that will essentially really clearly tell us whether or not these molecules have the opportunity to ameliorate the pathology of the disease in vivo. And then the steps after that, of course, are really then when we start getting into preclinical development of a drug. So we are moving forward. We are on track here in terms of the collaboration. We're totally on track with respect to the current milestones. And within a year, we hope to be at this point where we have the leads identified for this current project and be able to move to the next phase. So as I mentioned, we're optimizing active hit series currently to improve the drug-like properties, and then we're going to move those into some of the CMTA STAR network collaborator labs, such as Laura Feltry at the University of Buffalo, Gabsang Lee at Johns Hopkins, and then into animal models at Psychogenetics. So I just want to end and say, you know, this isn't done by one person, and it clearly takes a village to actually move this drug discovery forward. There's a photo here, and the individual with the dark sunglasses on is Paul Wright. He's the project team leader here, and he's been my key team member that's really been driving this project forward in my laboratory. And you know, we have a lot of people to thank here for helping us sort of move this forward, particularly the CMTA. Without you, this project wouldn't be happening. And so we are very grateful to what you've put in place to really enable us to help us discover a drug that will ultimately help you. So thank you very much.
Thank you. Thank you so much, Dr. August. Thank you.